Thank you, everyone, for joining our first seminar. So I'm happy to be the one introducing the seminar and our first speaker. But first, we would like to give you just a quick overview on the cell therapy program and what we intend or aim to do via this program. So the UC-CHLA cell therapy program, it's a new structure, which is a joint partnership between Keck Medicine and Keck School and CHLA, as well as the Norris Cancer Center. And our aim is to support and advance the development and clinical translation of cell and gene therapy. We also plan an integrated good manufacturing practice facility. It's a requirement for biomanufacturing and clinical testing of these therapeutics. This is currently being constructed at the Norris Research Tower. And we have a model that we would try to communicate with all our colleagues internally and externally, which is our aim to establish a partnership. We don't want to act as a contract manufacturing organization, but rather like we as a group of scientists managing this program, we also would like to partner with our colleagues in bringing their therapeutic concepts to the clinic, as well as offering training to next generation of investigators. And a very important component of the cell therapy program is our seminar series, which we are starting today. And this is, again, this is aimed to create a platform for deliberations and discussions on important topics related to translating biotechnologies in the most efficient, safe, and timely manner. We made an exception for today, given the busy schedule of our speaker. So we started we have it on a Monday today at 1 p.m. But following this, we will be first Thursday of each month at 12 p.m. And with that, I would like to introduce our speaker. But first, I think we had, if we go one slide back, so just some housekeeping instructions. Please wait until the end of the talk if you have any questions. And you have two options. Either you can raise your hand and you can address our speaker directly, or you can just type in the Q&A the question you would like to ask, and I will be happy to read it out loud. So our speaker, which I'm very excited and happy to have him joining us, is Peter Marks. And Peter is the director for the Center for Biologics Evaluation and Research at the U.S. Food and Drug Administration. The center is responsible for ensuring that safe and effective vaccines and biologics, including cell and gene and tissue engineered products, are available to patients and the most efficient quality and safe way. So we thank Peter for his time. I know you have a very busy schedule, and we appreciate that you're making the time to join us today. Thanks again, Peter, and it's all yours.
Thanks so much. Just going to get my slides up here. Of course, they disappeared in a moment. There they are. Okay. Okay, let me just share my screen now. Thanks very much for having me, Mohammed. This is it's really a pleasure. This is an area that I find extremely exciting. Let's see. Okay, I believe you can see my slides now. Yes.
Yes.
Okay,
yes.
Great. There we go. Okay, so what I'll talk to you about today for about a half hour is our efforts at FDA to facilitate the development of safe and effective regenerative medicine products. This title, regenerative medicine, it encompasses a lot of different things, but what I'm going to really be talking about are cell therapy products or genetically modified cell therapy products. Sometimes more is put in this basket, but I'm going to kind of stay narrow here. Just as by way of full disclosure, I'm a full-time employee of the United States government, and I don't have any relevant commercial interests to disclose. So, the bottom line here up front is that we're very committed at FDA to advancing the development of safe and effective cellular therapies to address unmet medical needs. And we're trying to do so by encouraging sponsor interactions with our center, by deploying all of our applicable development programs, which I'll tell you some more about, and by taking appropriate regulatory action when needed to protect patients. Although this is, to me, an extremely exciting area of science, in part because of the incredible complexity. As somebody who's trained as a cell biologist, I mean, cells are incredibly complex. They make single proteins look incredibly boring, and they make small molecule drugs look like nothing in comparison of complexity. So these are incredibly complex products that offer tremendous hope if we can understand their complexity and use them appropriately. Unfortunately, what has happened often is people have tried to jump ahead of having an understanding of these products and have used them without evidence of effectiveness, and that has led to some unfortunate issues, particularly in the area of stem cells. So what are some cellular therapies? Obviously, they're such a wide variety of products, everything from bioengineered skin to bioengineered blood vessels. In this case, this is an acellular vessel that was basically cells grown on a scaffold to leave behind a tube, bioengineered bladder. Now, interestingly, all of these things I've just discussed, none of them are yet approved, although they're in various investigational stages. The interesting thing here is the genetically modified T-cell therapies, such as chimeric antigen receptor T-cell therapies, actually have made it to approval. And there are a couple of reasons for that, but in part it's because it's very interesting, I think, in kind of reflecting that the ability to use our molecular biology and genetics to essentially force the cell into doing something is what has made this possible. Because oftentimes when we give cell therapy products, cells do whatever the heck they want, and understanding the critical quality attributes for many cellular manufacturing processes has been one of the true challenges. Getting there by the trick of molecular genetics seems to help in some ways, and that's why we now have 5 CAR T-cell products approved in the United States. They started back in 2017 with tisagen-lecleucel, then we had axicabtagene, then we had brexacabtagene. So we got through both ALL and then non-Hodgkin's lymphomas, mostly high grade, and then mantle cell lymphoma, and now we actually have a CAR T cell for multiple myeloma, and idacabtagene, and additional CAR T cell therapies. And these are pretty impressive therapies, although they are associated with significant side effects, and they have to be used carefully. So those are really, it just makes me, you step back to say, these are products that clearly have met all of our regulatory standards for safety and effectiveness. It took a while for them to get there. You might notice that, some of these products were in investigation since, for years before they made it to market. And that's because it took a while to be able to make them reproducibly and develop an understanding of the critical manufacturing parameters. So some of what we've learned there has to be applied to other cellular therapy products. What I want to do now is just take a step back and tell you about our regulatory framework for cellular therapy products in the United States. So these products are human cells, tissues, or cellular tissue-based products, and we call them affectionately HCTPs. And they're articles consisting of human cells that are intended for implantation, transplantation, infusion, or transfer into a human recipient. And they include a lot of different things, including skin, cornea, and the cellular products that I've just described, such as CAR T cells, but could also be mesenchymal stem cells or pancreatic islets. We regulate these based on our framework. And for those of you who aren't familiar with how FDA does its regulating, I just want to take you back and give you a primer, a 2-minute kind of regulatory 101. FDA can regulate cellular therapies, even if they're autologous cellular therapies, because of the Constitution and the Commerce Clause in the Constitution. It turns out that even if you are manipulating an autologous product, it's likely that you ended up using material that crossed state lines. And it's actually that Commerce Clause of the ability of the federal government to regulate interstate commerce that allows us to do this. The way, obviously, there's no cell therapy clause in the Constitution, but indeed in the Public Health Service Act, there are portions that apply to cellular therapies, and they live in sections 351 and 361 of the Public Health Service Act, although again, cellular therapies aren't called out quite that specifically. To get to that level of specificity, one has to go down to a level that is not something that's made in Congress, like our laws or statutes. Statutes are just another word for laws. FDA and other federal agencies make regulations or rules by a process called notice and comment rulemaking. First, we say, oh, we're thinking about making a rule about cellular therapies. That's called an advance notice of public rulemaking. Then we put out a notice of public rulemaking for comment. That's basically what we intend to put out as a rule, so people can comment on it. And then when we put out the rule, we put out a response to all those comments, and then it takes effect after a period of time. And that's how the Code of Federal Regulations that the FDA functions by was developed. We also, though, then have a layer that's even less of a I guess it's even expounds more on what we do at FDA, and that's called guidance. And guidance are not regulations. Guidance are ways that we at FDA say that you can stay on the right side of our regulations. So it tends to be things that are recommendations, but not mandates. And so when I'll talk about us issuing guidance on something, it's because we've issued something which says, here are some recommendations on how you can stay on the right side of our regulations. So the laws relevant to our HCTP regulation include Section 351 of the Public Health Service Act, which basically says that if you have a biologic product, you need to have a license to distribute in interstate commerce, that the products have to be safe, pure, and potent. And it also gives us this power to give licenses, take them away, and to also recall products if they're found to be have any issues with purity, potency, or safety. There's also Section 361 of the Public Health Service Act, which allows FDA to enforce regulations to prevent the introduction or transmission of infectious diseases. And the reason why these two provisions are relevant is because we regulate certain products just to prevent infectious disease transmission and not for safety and efficacy, whereas for other products, we regulate for both. And when we're not regulating for both safety and efficacy under Section 351, we use 361 because the safety and efficacy is taken for granted if you prevent the transmission of infectious diseases. So how does that work? Well, the rules for tissues became effective on May 25th, 2005 by the process I described. We developed two tiers of regulation for human cells and tissue products. One, in which these are typical biologic products. These are either drugs, devices, or biologics that are regulated just as other medical products. One submits a pre-market application with manufacturing information and clinical data. We review it, and you get an approval or a letter that says what you need to do to get it approved. On the other hand, there's a whole other category of products which we wouldn't ever want to have to go over and over again to have people come in for a biologics license application, because there are things that we do every day in medicine that are, by definition, through years of experience, safe, and essentially, because they've been done often enough, we take their efficacy to be granted, in part because in some cases it's apparent on face value, and in other cases what's being done is simple enough that it would be, again, okay on face value. And so those are things that are subject to the tissue regulations, which don't require pre-market review and approval. So what are these? Well, think about a skin graft. You wouldn't want to have to go to get an approval from FDA every time you wanted to do a skin graft, or even if you had allergenic skin that was going to be used perhaps for grafting every time you did this. And so we have a set of in our regulations, we have a set of criteria which say, hey, if you're doing things that don't require a lot of manufacturing, that is, they're minimally manipulated, they're going to be used like for like. That is, you're using skin to replace skin, or fat to contour fat like you would with fat, or muscle for muscle. Those are homologous use. They can't be combined with another article, with some rare exceptions. In other words, you can't have combination products here. You can't take cells and throw them on a scaffold and then call that something that could need this exception or could meet this exception. And then there's this fourth prong, which is they can't depend on the metabolic activity of living cells unless they're from a related donor. So why does this present problems for people sometimes? Those criteria are a little bit complex, and it does take a little interpretation to figure out what is truly minimally manipulated. In other words, what does not undergo significant manufacturing, because you could say that, well, I just sized the tissue some, I washed it, and I might have soaked it in a little antibiotic, but then I washed the antibiotic out. And that would probably be minimally manipulated. On the other hand, you might have done something different, like digested the tissue in enzyme, and so it's no longer has tissue integrity, it's just a bunch of cells, and then you let those cells grow overnight. Well, that's clearly not minimally manipulated. But then there are things closer in the center, like, well, I didn't use an enzyme to get the tissue dissociated, but I used a special type of grinder. So it can get complicated. And because of that, we put out guidance, which are our recommendations for how one can determine if they need, people need to determine if they need to get pre-market authorization for their products. And the other reason why we decided to put out a whole suite of guidances in 2017 was because we really wanted to help the field advance. Because what had happened was there was a lot of products getting used in this area for which there were no real data, yet people were buying them, people were paying for them, and we really wanted to see the work done to understand whether these products actually work and provide medical benefit, or whether they're really just very expensive placebos. So our suite of regenerative medicine guidance documents included one on something that I did not mention already, which is that one of these things is that certain things we really don't want to capture in our regulatory framework. When you as a surgeon take out somebody's, do a total thyroidectomy, and you take out the parathyroid glands and put some in the freezer so you can put them back later on in somebody's arm so that they don't become hypoparathyroid, we don't want to get you into our regulatory net. So basically, things that where you do things in the same surgical procedure, or when you move a vein from place A to place B in the body, that doesn't come under this, this regulatory framework at all. So we needed to define that. Our second guidance was one I'm going to tell you more about, which talks about how you define when things are minimally manipulated or used homologously. There was a guidance on devices, which I won't say more about, except to say that we try to expedite the development of these regenerative medicine devices as much as we can. And then we'll talk a little bit more about what we've done to try to help expedite regenerative medicine therapy development, both through a pathway that Congress set up called Regenerative Medicine Advanced Therapy Designation, as well as through some ideas for putting together development programs that can get there. So this issue on minimal manipulation and homologous use, our guidance explains that these are separate things. You can have something that's minimally manipulated, and if it's not homologously used, it still falls out, or vice versa. Some people used to conflate the two things, but they are distinct concepts. We also had to clarify that for our purposes, adipose tissue is a structural tissue for the purposes of applying our regulatory framework. Now, every time I give this talk, somebody argues with me about the fact that adipose does have a hormonal function. It also has nutrient functions and other functions. That's true, but for our purposes, we consider adipose a structural tissue because of how it works in general. It works, this helps us in our regulatory framework because by considering it a structural tissue, it allows us to, for instance, consider adipose something that's reasonable to use for breast reconstruction when it's moved from one place in the body to another. It's a structural tissue, and we don't get into the issues with action at a distance of cells transplanted. We also talk about in this guidance how the homologous use definition applies, because sometimes that is something that people would be questioning. Well, you know, there's skin, but what if you rolled up skin and put it beneath the skin as a filler? Could that be homologous use? Well, maybe not. But people have all sorts of questions like this, and so we clarify this in our guidance, and we clarify how to understand this. We also in this guidance explain that, you know, right now we're in a period where a lot of people have been marketing products, that's actually selling products for which they don't have efficacy information. In some cases, they don't even have safety information. Those include everything from amniotic membranes for this or that that are modified, or amniotic cells or amniotic fluid, which by the way is not an HCTP, but they market that. Also, there are various stem cells from various sources that are marketed. And what we needed to do is we wanted to make sure that people knew, look, for the legitimate actors who might have not realized that they needed to have an investigational new drug application and ultimately get a license to market these products, we wanted to give them time. So we originally were going to give them 3 years until November of last year, but COVID-19 came along, so we added 6 months to things. So at the end of this month, our period of enforcement discretion for these products will end. What does that mean? What that means is that if somebody is actually selling a cell therapy and they do not have a license, and that cell therapy is destined to do things that are not minimally manipulated and not homologously used, so they fall under our regulatory framework for needing pre-market authorization, we will have to take enforcement action, and we can talk more about that later. So this is basically in an effort to try to clean up Dodge, so to speak, so that we have therapies that are safe and effective. What we also did, though, that you might say is the stick, we also would like to have a carrot. And the carrot here is that we have the regenerative medicine therapy provisions from Congress, and we issued a guidance which described how we would apply these regenerative medicine advanced therapy designation, which applies to certain cell therapies, tissue engineering products, human cell and tissue products and combination products. We also, in this group, could be genetically modified cell therapies and gene therapies that give durable effects. Transient transfections that just give a brief expression of an antigen don't qualify, whereas things that lead to durable effects do. And so our regenerative medicine advanced therapy designation is for products that are intended to treat serious or life-threatening conditions. There has to be preliminary clinical evidence that these products can meet unmet medical needs. And we then review these very much in a similar manner to breakthrough therapy designation. And the benefits are very similar to breakthrough therapy designation, with two exceptions. First of all, the level of clinical evidence required is not as high. And second, once one gets an approval, if one gets an accelerated approval, there is a wider range of ways to fulfill post-approval commitments, and they include being able to use real-world evidence, expansion of existing data sets, using more patients into a clinical trial. So that can be helpful for the regenerative medicine advanced therapy developer. We also, in this guidance, discuss how to deal with this concept that's becoming one, and this is potentially applicable to CAR T cells, which is that more and more there are various academic institutions that say, hey, I want to make my product at my facility, and I don't want to be beholden to some company outside of our institution. Nonetheless, the institutions are not large enough to develop the data necessary to support a licensing application. So what we have described here, and this was described in a New England Journal paper by Scott Gottlieb and myself, is this concept that, our traditional drug development is you have a single entity that makes the product, they conduct a multi-center clinical trial, and they get the product approved and then distribute it. But you could imagine that you could have multiple facilities, each making a product according to a very set protocol with perhaps some quality control to make sure that they're all making a very similar, if not identical product. They then enroll patients into a multi-center clinical trial. Obviously, they can enroll patients at their site. And then each of these centers at the end is able to take the data from that multi-center clinical trial and use it to potentially get an approval for a license at their individual site. And thereby you have, okay, I'm not dependent on these other sites once I have my license. And the only real issue here when one goes down this route, which I think we may see some examples of this coming down the pipeline in the not too distant future. The only real important thing is that upfront one takes care of all of the intellectual property issues that one needs to, so one doesn't end up with arguments at the end over who did what. We have a program called the Interact program, which is an early regulatory meeting, which allows sponsors to come in to get informal regulatory advice very early in their development program so that you don't make missteps. And we strongly recommend this for cell therapy products because there are many things that we've learned over the years from seeing literally hundreds of cell therapy development programs that we can help people avoid missteps. Things like being able to say to people that they need to get a potency assay to understand a critical quality attribute of their product very early on so that when they get later on down the line, they can reproducibly manufacture their product. Giving that kind of advice really saves people a lot of grief down the line. So these are what these regulatory meetings are for. And I did send Emily these slides. You're welcome to them. And the link on here takes you to a standard operating procedure for how one goes about asking for one of these meetings and what one needs to submit, which isn't very substantial. So just to finish up, we're very committed to moving these cellular therapies forward. They're tremendously exciting. I think we still have a lot to learn. And a lot of this is in the area of manufacturing and consistency of manufacturing, because many of our problems here that we see are not in the area so much of clinical development. They're not absolutely related to clinical development. They're related to the fact that the manufacturing process that's been used for that clinical development is not reproducible. So we look forward to working on this with others, and I'll stop here and look forward to taking questions.
Thanks so much, Peter. This is excellent and very helpful. And listening to you, I can, I think, talk on behalf of some of our colleagues that sometimes regulation seems like a black box, that they are not familiar with, and usually some of these terminologies they haven't heard before, and when they get across or proceed with translation, they come across these terminologies and these issues, and they find it difficult to navigate. So this was very helpful to give us like a 101 on regulation and what is the FDA is doing. Thank you. I think until we receive questions, I would like to start by asking some of our colleagues here. I know that they are embarking on submitting an IND, and they have maybe a therapeutic concept in development. What advice would you give them while they are going through this process? What issues, pitfalls they should take care of and be familiar with before they submit their IND?
Right. So that's a really great question. So I'm going to say some just practical advice, and some of this you can discard if you'd like. First of all, if you're submitting something, try to find something that's already been submitted to the agency by somebody who's friendly to you, and get them to share what they've submitted. Because if they've got an IND in place that's been approved by the agency to proceed, and you can kind of mirror that in many ways, you're going to be in very good shape. You can't always get there, but to the extent that you don't want to copy paste, but if you can get good hints from what was successful in the past, it can really help you moving forward. Another thing is just remember, I think, so that's number one. Number two is just remember, we often underappreciate how important consistent manufacturing is. If one has consistent quality manufacturing, the agency is usually willing to take much more regulatory risk in first-in-human studies than when one has a very piecemealed, I'm trying to be very polite, piecemealed manufacturing process. So again, that's an important piece to think about. And then the final thing is, don't be bashful about using the tools of coming into the agency early and having that discussion, that interact meeting discussion early. Once you have a specified product, if you have concerns or questions about how you will move forward with the preclinical toxicology or the manufacturing or even the early clinical development. So again, it's copying something. It's not copying. It's getting good advice from someone who's been successful before is good. Understanding the importance of manufacturing, and don't be bashful about asking the agency.
Got it. Yeah. Thank you, Peter. This is helpful. So we have a couple of questions here and also a raised hand. So I will start with the questions that came first. So how will the FDA approach the regulation of gene editing for very rare pathogenic variants in EQUAL1?
You know something, whoever that was such a nice plant. If you planted that question, you couldn't have done better. Thank you so much. We are currently working on pathways. We don't have it all worked out, so I'm not going to lie to you and say everything's worked out. But we're currently working on a kind of a schema for how one can potentially do this. And the idea here is to use what is essentially an originator product, for which it might not be used in just one person. It would be used in multiple people, for which you have data on the manufacturing potentially and clinical data. So you feel comfortable that that product is as could be a reference. Then you'll have offshoots like these N of ones, which you make in a very similar, you use the same manufacturing technologies. You can leverage that way, that preclinical tox, et cetera. And the idea then is that you could have all these offshoots from one originator or one reference. I try not to use the word reference product because we use reference for biosimilars. So I'm trying to avoid that. So what I'm lapsing into is the word I try to use originator and offshoots, but essentially what we're doing is taking a reference product and then having subsequent products look back at that so that you don't have to go through and reinvent the wheel every time, because you're exactly right. There's going to be rare diseases where it's going to be N of ones all the time because the genome editing will be that way.
Perfect. Thanks, Peter. So this question was from Ryan Schmidt, our colleague here. Another question from Debbie Metra: what is the benefit of breakthrough designation?
Yeah, another great question. So breakthrough therapy designation allows one to, once you have breakthrough therapy designation, it essentially automatically qualifies you for use of other of the expedited programs. And almost all breakthrough therapy designations get priority review, which means you get your application reviewed essentially 4 months sooner by the agency when it comes in for a license or a new drug application. But even more important than that, when something is breakthrough designated, it comes up the agency to the attention of senior management, and it has the ability to have more meetings, because if something's got breakthrough designation, it means that the agency thinks that it is enough of an advance over existing therapies that it should take our additional effort is worthwhile to make that product move forward more quickly. So in general, it means having more meetings and having senior agency staff know about the product, which can be helpful because sometimes we're able to help move things forward a little bit more quickly.
Yeah, so early interactions and involvement more from the FDA, which is very beneficial in the long term. So third question, is GMP manufactured product required first before an IND will be approved?
Yeah, so this is a really good question. And this one is one that I should say, so it depends a little bit. So if you are a academic investigator, it can be GMP-like potentially for early trials, but you'll eventually need to switch over to true GMP product. Now, if you're a commercial manufacturer, a commercial entity, you should expect to be doing GMP from the start. It's true that because academic investigators, sometimes they're treating very rare conditions, and we don't want to prevent someone whose life could be saved from standing on ceremony that somebody did not have every last piece of GMP in place. But in general, though, as you progress your program, the GMP requirements will increase until you have a fully GMP compliant product as you come into later stage clinical trials.
But just to make it more clear also to our colleagues, like we need still basic requirements in whatever product we are giving to patients.
Yes. No. That's really important. That's what I was just trying to say. What I was trying to say is we can go GMP light, so to speak, but it can't be, it is true. Occasionally we get a manufacturing section from somebody where it basically says, we're going to isolate some cells, culture them overnight, and make sure they're not contaminated and give them to somebody. That probably won't suffice as the manufacturing section. On the other hand, a good detail of where the cells are coming from, how they're being processed, what are the controls that are being done, what tests are being used to ensure sterility, that will get there, even if some of the issues show that it's not a fully GMP qualified cell bank.
Yeah, thank you. I think this is important, especially like when we compare between academic and industrial processes. We talk more also about potency in advance, having in place a good potency assay. Sometimes academics wouldn't have this very well established, characterized
Yeah.
at this stage.
I will tell you, if you want to add value, for those tech transfer people who might be on the call, if you want to add value to a manufacturer or a sponsor who's looking to tech transfer a product, if you have a potency assay that you can show is a good potency assay for that product so that you can make it reproducibly, you have an incredible value added. So it's a good thing.
Yeah. Thanks, Peter. So Dennis, please go ahead, ask your question.
It's Dennis Usenko from Orthopedics, USC. We have developed an implant for focal articular cartilage lesions from embryonic stem cells and have successful pre-IND meeting for that with FDA. But in general, embryonic stem cells, they have been a big ethical concern for a long time. So in California, we are privileged to have California Institute for Regenerative Medicine supporting us. But what's the FDA's mid and long-term view on cell therapy products from embryonic stem cells, human embryonic stem cells?
So yeah, so generally on these issues, we try to stay neutral. We go with where Congress tells us to go. The good news is Congress and the department, it sounds like I can't promise anything, but the latest I heard, it sounds like things might be headed in a more permissive direction under the current administration than they were under the previous administration.
Okay, thank you.
Yeah.
Go ahead, Peter.
It's a good question.
Yeah. Next one from Ian Hutchinson. So some cell-based therapies, such as CAR T cells, have both serious and predictable adverse events. Would the FDA mandate they have to have a successful treatment protocol? Would you insist on inserting a suicide gene, for example, for CAR T cells to switch off adverse events?
Yeah, really great question. So when the first CAR T cell came around, we had these really bad effects, and it was lucky that there was, the IL-6 inhibition, tocilizumab, which led to remedying some of this. If you had, so as long as you have something, it would all depend on what you have and the extent and how severe it is. Right now, for the CAR T cells that are in place, most of them are not relying on suicide genes. They're just relying on treatments that can be given after the fact if tumor lysis syndrome or essentially this essentially cytokine response syndrome comes up. The neurologic toxicity is so idiosyncratic, it's hard to know what that would help with for that if you had a suicide gene. But as we see newer, more potent CAR Ts coming up, we're starting to see more and more. It's not just one, there are multiple people trying to put in suicide genes, with the idea being that if things get too hot, you could then cool them down quickly. We don't mandate it, but what we do mandate is that the benefit risk when the cells are administered be reasonable. If you had something that had the potential for a very life-threatening toxicity that if could not be controlled by any other means, then perhaps you would want to have a suicide gene in your CAR T. But it's one of these things that we risk-benefit judge.
Yeah, thanks, Peter. I think if I may add, I think my understanding is that the FDA would look at any risk mitigation measures that the investigator has added to the clinical treatment plan or the clinical trial, and they evaluate this potential risks versus whatever measures they have in place, maybe advise them to add more risk mitigation to avoid any kind of like expected adverse events. And this is how
Right.
the decision is usually being.
Exactly. Exactly.
We have a question from Dove. I think it's related to, I'm going to sum up instead of reading the entire question. So I think it is related to feeder cell lines or cell lines of human origin and their use in manufacturing a cell therapy product, and whether they should be or not incorporated in the final cell therapy product.
Yeah. So this is one that gets to be complicated, and I would say this is one you come into the agency and have a chat about, because it may be okay, it may not be okay, depending on the specific circumstances. So have a conversation about that one.
I think it's a case by case.
I don't want to give you bad advice on that one, because I could imagine I could answer it both ways, and it would all depend.
I think it's a case by case, like you said.
Exactly.
So, yeah.
Exactly.
So another question, like in your slides from Connor, in your slides, you mentioned the new multiple facilities development strategies. And the question is, what type of SOPs variation is acceptable in the manufacturing if to be considered the same product?
Yeah, so really, this will all depend on what you have as critical quality attributes. So if you have multiple critical quality attributes that you can measure on your product and show time and time again that those are the same from different sites, one could possibly tolerate small variations in SOPs, because maybe it was more convenient at one site for the cells to be incubated overnight, whereas others during the daytime or something. Whereas if you don't have good control of your critical quality attributes, then you're going to probably have to be stuck with doing the protocols very similar, as close as you can get them from site to site, because you don't have something to rely upon to, at the end of the day, say you did the same thing. So it really does depend on the robustness of your your quality attributes or potency assay, et cetera.
Exactly. Thanks, Peter. So next question from Young Mikem. Great talk. What is the success rate of pre-IND filings, and what is the major reason of rejection? And
yeah, interact meetings. Yeah, this is a great question, and there's an asterisk here. Right now, we're probably for our interact meetings only granting somewhat less than half of them. Whether it's a third or so is probably right. The asterisk there is we very much want to, in general, be able to grant almost all of them where there's enough information. The problem is with COVID-19, the agency has been overwhelmed. We have vaccines coming out of our ears, cell therapies coming out of every other orifice. So we're just trying to keep ourselves above water. But one of the things we're very committed to is, as we move into the next set of user fees, and as we get staffed up, to try to be able to do this, because it's my personal belief, and one of the legacies I want to leave with our center, that having interactions very early on and getting things off to a right start, there's nothing that matches that. If you're off to a good start from the beginning, yes, things can go wrong, but you've started with necessary and a necessary beginning. On the other hand, if you get off on the wrong foot, it can take a long time to get back on track.
Thanks, Peter. So Michael Myers is asking, would the FDA allow a much improved CAR T-cell therapy trial to start without having head-to-head comparison to the existing approved CD19 products?
Yeah. So a great question. And in general, for these products, we've allowed single-arm studies if they are clearly, because they're biologics, we can allow single-arm studies because they're not really, we're not comparing exact to exact, and we understand that. So, and that's why if you look, there are 1, 2, 3, I think I'm right, 3, 1, 2, 3. Yeah, 1, 2, 3. There are 3 CAR Ts against anti-CD19 CAR Ts because they're all slightly different. You can certainly come along with that. So that's a perfectly legit pathway for biologics.
I think Breed Chaudhary, you have a question. You raised your hand. Go ahead.
So my question is, I also put the question in the chat that what kind of preclinical safety studies are needed to demonstrate that the antigen binding domain doesn't have off-target activity, because most of the antigen binding domains are unique to human antigens. So,
Great question. So this is gonna depend. You will probably likely need at least one species or two species potentially, depending on what the target is and how well we understand that target. One of the reasons why CD19 has been so favored is because we understand so much about it. The same thing now with BCMA. But as we go into other targets, this becomes another issue to have to discuss with the agency, because you're spot on right, especially as we come to some of the people trying to address solid tumors. There can be off-target effects, and that's why some people are making these Boolean logic CAR T cells using genome editing in part, where you basically have, for instance, an on signal, something that identifies it, but it's also an off signal where it is, which is dominant, which is if it sees that that other molecule on the cell surface, it says, no, don't activate. So these complicated constructs are something that's being put in place, but it's a great question. The preclinical tox will depend.
so let me, if I may ask a follow-up question. So let's say that there are now mesothelin, CARs and, PSMA CARs and, HER2, of course, we have a lot of experience with Herceptin. So if we want to target these antigens, which have been already been validated to some extent using either CAR T cell or antibodies or ADCs, do we need to then do any primate studies, or mouse studies are going to be enough?
I'm not going to speak definitively, but you probably would be because the cellular therapy aspect of this is so much more potent than the antibody itself that you might uncover things that you might not know otherwise. So you'd probably need at least some toxicology study. But again, if you're really interested in that, I would have a conversation with our office just to make sure that they'll make sure you're set on the straight and narrow with that.
Thanks so much, Peter. I think if I may add, it's always like a matter of what existing evidence that you can use to support your
That's right.
argument, and how much you can convince the FDA that you only need to do minimal toxicity.
Right. I think the Herceptin is a great example. There, because we're so cognizant of where things are and knowing, as long as you didn't have an issue with the heart getting a little tickled, but this is there's some of these antigens are more well defined than others.
Yeah. So Yang Chai is asking if we have a device for tissue regeneration that will require FDA approval. We also want to load autologous bone marrow as aspirates onto the device. If this is still a device-based or review-based review approval process, I think does it fall within the parameters of?
It falls within CBER. And in some cases, it might be a biologic product or it might be regulated as a device, but it doesn't matter because we'd regulate it either way. And we will always try to regulate it using the least burdensome regulatory pathway. We don't get extra bonuses by doing something more, higher regulation. We don't get any bonus for more regulation.
Yeah, and another question is asking about your current thinking, FDA current thinking on allergenic versus autologous development in the preclinical studies requirement, and particularly the point of immunogenicity evaluation in cellular therapies.
Yeah,
I think you cannot play favorite, but of course, again, it's a matter of data and how much you can generate in advance.
Yep, no, that's exactly right. I'm going to have to drop off in a minute because I have to, I'm sorry, I was a minute or two late, but I do have a
Yeah,
5 o'clock that I'm going to have to get to.
Okay.
But one thing that one can say is that I see a question from Yiben, and about, one thing I would say is it is true in increasingly, and it's almost perfect circle because it goes back to the N of 1 therapies. For genome editing, there is no right animal model because if you have to use a human genome sequence to target, using a mouse to do, unless it's a humanized mouse where you're targeting the humanized mouse with like a humanized liver, you can't know that you're doing things right. So we're increasingly looking at organoids, other models to try to get to that.
Perfect. Thanks so much, Peter. I know
Thank
we are
you
running
so
out of
much. It was
time.
fun.
Thank you. And we may send you some questions that we can answer via email and share it with our colleagues.
Please do. My
Okay.
pleasure. Take care.
Thank you. Take care. Bye.
Bye.